Hi, everybody, and welcome. While everyone keeps filing in, we'll just start with some housekeeping. So it's great to see you all here today, and thank you for joining us in the space for the Stream webinar series. So I'm joining you today from North Vancouver on the unceded territory of the Coast Salish peoples, including the territories of the Musqueam, the Squamish, and the Tsleil-Waututh nations. So we have a chat box, so please let us know where you're joining from today and take a minute to introduce yourself, what organization you're with in the chat box. Amazing, it looks like we have people from across the country. So welcome. We have Nova Scotia, we have British Columbia. Thank you for introducing yourself. Newfoundland, welcome. Thank you. So before we get started, please note that there is also a question and answer box, and we will have a question and answer session after all the presentations have completed. So please take a moment and find the question and answer box in your Zoom controls. So we're excited to come together for our first webinar of the Stream series today, hosted by Living Lakes Canada. So before we get started, I'll just share a little bit about Living Lakes Canada. So Living Lakes Canada is a national nonprofit organization, and we're working towards the long-term stewardship of Canada's fresh water. And so our mission is to normalize water stewardship through community-based monitoring. And we believe that this is a way to empower localized climate adaptation and provide support for decision-making in filling important water data gaps. So much of our work has become a grassroots template for aquatic ecosystem monitoring across BC and much of Canada. And we're also affiliated with Living Lakes International, which is a network of over 120 non-government organizations across the world that share the same mandate as us, working towards the protection of lakes, rivers, wetlands, and watersheds internationally. And so we have a number of projects on the go at Living Lakes Canada that you can see up on the screen here. For more information about any of our other projects, please visit our newly updated Living Lakes website. So just a reminder that this is the first of a series of 4 webinars. And you can see the dates and the times listed for the next ones there. So please register for those as well. We're really excited about a roster of speakers. We have a great lineup for everyone that'll be joining us over the next few months to share their experiences with the Stream projects and their monitoring programs. And so the goal of this series is to provide viewers with an overview of Stream, what we have achieved to date, the outreach that we have done, and also just introduce you to the fabulous community-based water monitors that we have partnered with to make Stream what it is. So it should be a great series, and we're really excited to share it with you. If you have any questions, we invite those viewers to get it, and you're interested in becoming involved, please reach out to our team. Okay, so today's webinar series kicks off with the presentations from representatives from the core Stream project teams. And so this includes Dr. Chloe Robinson with the University of Guelph, Catherine Paquette with World Wildlife Fund Canada, Adam Martens with Environment and Climate Change Canada, and myself, Reagan Mallinson with Living Lakes Canada. So first off is Dr. Chloe Robinson with the University of Guelph. And Chloe is our projects manager for the Stream project, and Stream stands for Sequencing the Rivers for Environmental Assessment and Monitoring. So it is an acronym. And the Stream project is within the Center for Biodiversity Genomics. And Chloe's research background is largely in molecular ecology, with a focus on developing novel environmental DNA techniques for monitoring of aquatic biodiversity. During her PhD, she worked with numerous community groups to collect environmental samples across the UK. And so Chloe is interested in the use of eDNA as a non-invasive method for monitoring of rare and endangered species and ecosystems for conservation. Chloe has an extensive background in science communication and has delivered presentations in a number of UK-wide science outreach and competition. In the STREAM project, Chloe is responsible for liaising with partners and community groups to collect samples from across Canada. She performs the analysis of environmental DNA and produces the DNA reports and scientific papers. So take it away, Chloe.
Thank you very much. So welcome everyone. I'm calling in from Guelph today. And Guelph is located on the ancestral lands of the Attawandaron people and the Treaty of the Lands and the Territories of the Mississaugas of the Credit. We recognise the significance of the Dish with One Spoon Covenant to this land and offer respect to Anishinaabe, Haudenosaunee and Métis neighbours as we strive to strengthen our relationships with them. So today I'm going to be speaking to you about the STREAM project and giving you a little bit of background in DNA metabarcoding, which is the main technique we use in STREAM to generate the data. So I'm not going to go over this information too much because I know Catherine will be covering a lot of the Watershed Report information. However, I just want to give you a bit of background and set the scene as to why the STREAM project came to be. So Canada holds 7% of the world's renewable fresh water. However, as you probably know, a lot of these freshwater systems are currently experiencing high threat and pressure from a number of means. For example, over abstraction, pollution, invasive species, habitat loss, and the list goes on. Across Canada, this is causing aquatic habitat degradation at quite significant scales. And to the extent of we can't actually determine the effects of these threats because many of Canada's watersheds are data deficient. For example, for benthic macroinvertebrates, 64% of watersheds are data deficient. Therefore, obviously, this shows that there is still this need for widespread data generation, particularly for these taxonomic groups. So I mentioned benthic macroinvertebrates on the last slide, and to give a bit of background as to why this taxonomic group is important, they are sediment-dwelling invertebrates. They are often the larval stage of flying insects, and they have different levels of tolerance to environmental conditions, such as water pollution. So some families or species can be incredibly tolerant of very, very poor water quality, whereas other families and species can be incredibly sensitive to poor environmental conditions. Therefore, they act as bioindicators for freshwater health. And the groups we target for this include caddisflies, mayflies, stoneflies, dragonflies, and damselflies. One way to collect information on benthic macroinvertebrates is to collaborate with local community groups through community-based water monitoring. And this has really revolutionized freshwater data collection, especially across Canada. And a lot of this work has been done through the Cabin network, which again, I won't go into detail because I know Adam will be speaking about this soon. And we're starting to see this transition from solely morphological analysis of benthic macroinvertebrates to a more DNA-based analysis. And essentially this involves processing DNA from environmental samples and matching it back to specific libraries to identify which species are present in the environment. And essentially this is how the STREAM project came to be. So as Reagan said, STREAM stands for Sequencing the Rivers for Environmental Assessment and Monitoring, and it's a Canada-wide community-based monitoring project. The main premise of STREAM is to provide a rapid turnaround and increase throughput of freshwater data. So this is looking like report generation from sample submission to getting the final report around 2 months, where possible. And obviously we have down to this species level taxonomic identification, which isn't often able to be achieved through morphological means. So the STREAM project works by training and working with community groups via the existing Cabin water monitoring network. And data is collected directly from bulk KICnet benthos samples by community-based monitoring groups, with the aim of collecting between 1,200 and 1,500 samples across a range of Canadian watersheds. The main overarching goal of STREAM is to validate DNA metabarcoding as the mainstream approach to be routinely implemented by groups such as Environment Canada, WWF Canada, and River Lakes Canada for generating biodiversity data for freshwater benthic macroinvertebrates. Some of the outcomes of the STREAM project will obviously be the individual community reports. So to date, we've sent out around 50 community reports since the project started in the end of 2018. And essentially, these community reports will end up feeding into a big 3-year watershed report to look at the overall health of watersheds across Canada. One of the really great legacies of this project will be trained water stewardship groups. So these are groups that are trained to collect data beyond the lifeline of the STREAM project. And of course, overall, better understanding of freshwater health is one of the major outcomes of this project. So STREAM does not function alone. It is a nice combination of Living Lakes Canada, WWF Canada, Environment and Climate Change Canada, and the University of Guelph, in particular the Haji-Babai Lab, which is where I'm based. And this slide here shows the collaborative workflow where all the different organizations are involved in the process of the STREAM project. So one of the most vital parts of this is rapid report generation and effective result dissemination to community groups to then feed into the following year's sampling and recruiting more community groups and training more people in environmental stewardship. So I mentioned that the STREAM project is branching into these DNA-based methodologies for collecting biodiversity data. And the main approach using the STREAM project is eDNA metabarcoding. So eDNA is basically DNA that's left behind by organisms in the environment. So this could be aquatic environments, so in the rivers, it could be in the water, it could be in the sediment, but it's also persists in soils. But obviously for the STREAM project, we focus on aquatic eDNA. And there are two main methods of collecting eDNA. We can use bulk tissue, which is what we do with the benthic kicknet samples, where you're physically collecting the bugs themselves, but also any sort of shed DNA is also included in that mix. Or you can collect directly from the water. And again, we use DNA metabarcoding in STREAM, which I'll describe in the next couple of slides. But you can also use single species approach. So you can just say target the species of American signal crayfish in samples, or you can target wider taxonomic groups, for example, benthic macroinvertebrates, as we do. And essentially, this approach is kind of earmarked as biomonitoring 2.0, which was developed within actually the University of Guelph with collaborators as part of a large project up in the Peace Athabasca Delta. And this really demonstrated how DNA sequencing technologies can create this revolutionary biomonitoring approach by which we can get down to species level taxonomic identification across a wide range of sites and samples and within a fraction of the cost and essentially more reliable because we can produce these percentages of certainty to go along with the classifications. So used in conjunction with morphology, this method is actually incredibly strong. And the two methods combined together are actually a really great approach to generating widespread biodiversity data, which is currently what we're doing in the STREAM project. So here I just describe the morphological approach with the DNA metabarcoding approach. So essentially, they both start with an environmental sample. The main difference being that individual specimens are sorted in morphological samples and identified via microscopy analyses. Whereas in the DNA, we process and extract DNA from those samples. We then produce sequences from that DNA and use bioinformatic pipelines with reference libraries to identify species. But again, as you can see here, these two methods feed into sort of one greater good, if you may, for identification of bioindicators, diversity analyses, for reference modeling, which we're using in Cabin, and some network analysis, such as food web analyses. So the STREAM project itself has a number of outputs to community groups. The main one being the custom DNA reports. So these reports include taxa lists to both genus and species level. Again, as I said, it produces a percentage certainty of these classifications. Groups that collect samples over multiple years also get multi-year biodiversity comparisons, so we can easily rerun data from previous years to see if the libraries have improved somewhat to improve numbers of classifications from sequences. And we can also do additional interpretation, for example, look at odonates in samples, do a bit more analysis on families or genus level. We can look at disease host detection, for example, Whirling disease, and we can also screen and highlight any macroinvertebrate invasive species and provide some interpretation on that. So as I said, a really key part of STREAM is the outreach and dissemination. And we do this over a number of platforms. So we are on social media, on Twitter and Instagram. We produce science communication articles. We recently produced an article for called Conversation Canada that was picked up by the Canadian Geographic. I can add a link to that in the chat after my presentation. We also produce publications and we write blogs both on the STREAM website, and also WWF and Living Lakes Canada also produce blogs. And our website is actually a really good resource. And again, our link, there is a link at the end of the slide, but I can also put a link in the chat where we have the blog. We have some outreach materials. We have an interactive FAQ, photos, and soon we're going to have an interactive map of where all the samples are being collected to date. Here's just a screenshot of where our samples have come from the last 2 years. So we've collected a total of 821 samples with community groups since the start of the project. And here you can really see the wide range of locations that these samples have come from. And you can also see areas where we would potentially like to target going forward for the project and where there are still some existing data gaps. So thank you very much for listening. I'd just like to acknowledge members of the Hajiba Lab for all of their work and support, and the funding organisations, obviously Genome Canada and Ontario Genomics. Thank you very much. So there will be an FAQ. There is an FAQ, so please place questions there, and we'll have the panel at the end. And here are just some of our social media handles, our website, and our email address. Thank you very much.
Thank you so much, Chloe. Fabulous presentation, and a big shout out to Chloe is that she actually draws those macroinvertebrates that you saw on her presentation. Up next, we have Adam Martens with Environment and Climate Change Canada. And Adam has been with Environment and Climate Change Canada for 15 years with invertebrate taxonomy. Oh, sorry, he has 15 years of invertebrate taxonomy. And he's been involved with the Canadian Aquatic Biomonitoring Network since his studies at the University of New Brunswick. Cabin fieldwork has brought him to all corners of the country, and with the recent launch of the STREAM project, Adam has completed multiple seasons collecting DNA benthic samples, streams, rivers, wetlands, and lakes. He's kicked them all. So off to you, Adam.
Thanks, Reagan. So hi, everyone, and thanks for attending. So like Reagan, I'm also located in North Vancouver. So I'm joining you from the unceded territory of the Coast Salish peoples, including the territories of the Squamish, Musqueam, and Tsleil-Waututh nations. So my portion of the webinar is going to focus on Environment and Climate Change Canada's role in the STREAM project, a brief introduction to the the Canadian Aquatic Biomonitoring Network, or just CABIN for short, and the reason why we are interested in DNA. So CABIN delivers a methodology to characterize benthic macroinvertebrates broadly and consistently across Canada to support assessments of aquatic ecosystem health. A strength and success of the CABIN program is that it is built upon a collaborative network of networks, including multiple levels of government, NGOs, community groups, Indigenous organizations, and industry. Some of the tools offered through CABIN include the standardized methods, like the field manual and the laboratory methods that you can see below, a shared database, a website, analytical and reporting tools, and online training and certification. So one of the cornerstones of CABIN is the training and certification program. It consists of a series of online modules, which students take at their own pace, and a 2-day field course. And we have held field courses in various regions of the country. You can see Baker Lake, Nunavut, just in the picture below. So once trained, CABIN users gain access to a suite of web accessible tools. So the data management system, the analytical software, and reporting tools for the assessment of ecological condition for your sites. We have a more in-depth look into CABIN on the next webinar in the series. For those of you that would like to learn more, be sure to tune into that. So Environment Canada has a pretty long history of applying DNA methods with hopes that it can be a useful tool in aquatic bioassessments. And I've had the good fortune of hanging around long enough to be there from the nearly the beginning. So for my part, I became involved as this fresh-faced graduate back in 2008 when I participated in a BioBlitz in New Brunswick. A BioBlitz also is just a focused collection of insects in an attempt to record all living species in the area. So that's me in the front there, spending my summer pinning caddisflies for the collection. We collected adult aquatic invertebrates, so the flying kind, like you can see the dragonfly and the stonefly there. We had experts identify them and then sent them to University of Guelph for DNA barcoding. So we used a variety of collection methods. So we had nets, we used light traps on the left, and the Malaise trap, which is the tent-like structure, and that just intercepts adults emerging from the water. This effort really helped expand the existing DNA libraries, and it furthered the development of DNA barcoding process in Guelph. So then in 2015, I joined in an existing Environment Canada study in the Peace Athabasca Delta. So it's a beautiful UNESCO World Heritage site located within Wood Buffalo National Park in northern Alberta. The project was associated again with the University of Guelph, and we collected 138 paired DNA and morphological samples over a 6-year period. And this was to demonstrate the DNA sampling methods and highlight the increase of invertebrate community data achieved through the DNA metabarcoding process. This was a really important case study, not only for processing the DNA metagenomic samples in freshwater, but this also became a baseline protocol. So it refined the cabin method to account for DNA collection scenarios. And in the picture, you can see an Environment Canada researcher collecting a sample from the wetland, wearing gloves to protect against DNA contamination and carefully walking through some mucky bottom. So now with the involvement in the STREAM project, Cabin and Environment Canada are exploring the potential for nationally standardized protocols as an additional tool for aquatic ecosystem assessments. And over the past 3 years, we have collected samples using the cabin field methods as a baseline, which you can see in the bottom corner, with the adjustments made to minimize DNA contamination in the STREAM DNA collection method. And one of our main questions is, what can incorporating the DNA metabarcoding into the cabin program mean? So on the left, we have our current morphological identification track. So samples are collected in the field using the standardized cabin procedure, and then they're sent to a taxonomist. They will sort, identify, and count a subsample, and it could take a long time, and a matter of months sometimes, and I can personally attest to that as I've been on that end. The results you get out of the morphological analysis include identification to family level and sometimes even to genus level if your taxonomy contract allows. You also get an estimated abundance of the sample, and because you're taking a subsample only of that benthic sample, you can then extrapolate to the entire sample as to how many insects you have in there. So all in all, this process could take months to complete once all the analysis and reporting is finalized. And then on the right, we have our DNA metabarcoding sample. So that is collected using the cabin protocol as a baseline with the revised DNA considerations. So once it's collected and preserved, it's sent to a DNA laboratory, which processes the entire sample, and a list of present taxa is generated to genus and species level, or as the barcode libraries allow. This process can be much quicker once a sample is received, and data can then be analyzed and reported in a matter of weeks. So some of the benefits with the DNA metabarcoding process include a potentially shorter time frame in terms of the sample analysis. So you can see 6 months versus 2 months. Lower cost related to running many samples in a single sequencing run, and expanded and potentially more representative diversity measures, as the DNA method uses the entire sample in the analysis, whereas cabin traditionally uses just a subsample. A couple challenges in the process, which are quickly being resolved. are the potential for DNA contamination in the sample and gaps in the DNA libraries as they're still being populated. A few publications which have come out of the project so far include the application of DNA metabarcoding to biomonitoring and the DNA benthic macroinvertebrate sample procedure in a full version, and you can see here in a picture reference guide. And that's really helpful for people who haven't been out into the field for a while and need a refresher on the different decontamination processes of the gear and equipment. The fact sheet is especially helpful if you have any outstanding questions about the project or about our role in the project. And they include questions like, how do you collect the DNA samples? How are the DNA samples analyzed? What is the difference between the conventional taxonomic data and taxonomic information from a DNA sample? And will Cabin adopt the use of DNA for identification of benthic macroinvertebrates? So I will have, there's a link on the bottom, and I will share it within the chat afterwards where you can find this fact sheet. And then currently, the Cabin database is being populated with all of the DNA data that's being processed in Guelph in its own study, and analytical tools are in development. So after completing the proper training modules, users will be able to access their data as well as all other partner data in the stream project. And just a quick plug to the database. It now features a search taxa function, complete with images from the National Cabin reference collection, which you can see below, and some interactive maps of where the taxa have been found in all cabin samples across the country. And with that, I'd like to thank all my colleagues in the department for their hard work and support, and our various stream partners as well. So thanks for attending, everyone.
Thank you so much, Adam. Those are great photos. I encourage everyone to go check them out on the link that Adam provided. So up next is Catherine Paquette with WWF Canada. And so Catherine joined WWF Canada in 2014 as an analyst and is now a freshwater ecosystem specialist on the restoration and regeneration team. She is a project manager for Watershed Reports, which is the first national picture of health and threats to freshwater in Canada. And Catherine has led multiple iterations of the project, including the most recent relaunch in 2020. Catherine's responsibilities for STREAM include identifying priority areas using the Watershed Reports, identifying and coordinating with groups and organizations interested in participating in the STREAM project, and reporting back to these organizations with the results. She has spoken at multiple events, including the National Monitoring Conference, the Society for Freshwater Science Conference, the North American Lake Management Symposium, and was a plenary speaker at the Canadian Conference for Fisheries Research. She holds a Master's of Science in Physical Geography from the University of Ottawa. So, okay, are you ready to go, Catherine?
I'm ready to go.
Dive in.
Thanks, Reagan. Hi, everyone. So yeah, like Reagan mentioned, my name is Catherine Paquette, and I'm a freshwater ecosystem specialist with WWF Canada, and I'm based on the unceded territory of the Anishinaabe on the shores of the beautiful Kitchissippi River, also known as the Ottawa River in Ottawa, Ontario. And today I'm going to talk about, obviously, WWF Canada in our involvement in STREAM, so how we came to be a partner in this really cool project, and really how we use our watershed reports to guide that involvement. So first, watershed reports. So I'm sure some of you might be already a little familiar with watershed reports, but they were really the first national picture of the health of and threats to freshwater in Canada. And they're really this innovative and standardized way of saying whether a given watershed is healthy or not, whether it's threatened or not, and what those different threats might be. So it was initially published in 2017, like Reagan mentioned, and late last year in October, we re-released an updated version with any sort of new data that we could get our hands on. And this assessment uses 4 indicators of health that we see on our screen right now, and also 7 indicators of threat. So threat are things like pollution, habitat loss, climate change, and things like that. But today we're really going to focus on the health, because obviously one of those indicators are benthic invertebrates, and that's what STREAM is all about. And so for us to be able to assess the health of flow in Canada, or assess the state of water quality in Canada, we need data. And so our data comes to us from all sorts of organizations. And we don't use, we're not ourselves a monitoring organization. Nobody at WWF really goes out, to stream or to a lake or to wherever and monitor. So we really rely on the amazing network of water monitors across the country. And so we get our data from the federal government, we get our data from community-based monitoring organizations, from academia, sometimes even from industry. So really, we'll take any data that we can get. And so here we see the benthic invertebrate results from our last launch. And I realize I forgot to include a legend, but in this case, red is poor or very poor, and blue is very good health. But what should stand out to you when you look at this map is the gray. There is a lot of gray on this map. And what that tells us is that it's data deficient. We just don't have access to the data that we need to confidently attribute a health score. So we don't know if things are good. We don't know if things are not so good. In some of these areas, that might be expected. This is a very big country, very sparsely populated in some places. So in the far north, we expected it to be data deficient. But when we look closer at this map, we can see that around the Great Lakes, in parts of the southern prairies, areas where there's a lot of resource extraction or huge dams, we don't know how things are doing. And that's particularly concerning. And so I'm not going to dive into all the results. This isn't a presentation about Watershed Reports. But if you do have any more questions, please feel free to follow up, or you can visit our website as well. And we've got a cool interactive map, and you can find out how things are in your own neck of the woods. So following the release of our first Watershed Reports and our second Watershed Reports, we created these key learnings and these recommendations. So in all, as an organization, we've been working on Watershed Reports for almost a decade. And so we've definitely learned a thing or two about how water is monitored in this country, how it's assessed, how it's shared. And we really felt that this, that we can impart some of the things that we've learned. And so a lot of those key learnings are about how we manage our fresh water, how we collect it. One of the big challenges we had was the different monitoring protocols across the country. People weren't looking for the same things the same way, which makes it really difficult for data to talk to each other. But one of the big ones was the community-based water monitoring, so CBWM. And when we were doing our outreach to collect or to get as much data as we could, we connected with amazing groups from across the country. I'm sure some of you are on the call, and who are doing amazing work. They're going out, they're monitoring the data, monitoring their waters. They know what's going on. They have more knowledge than anybody else about their particular river or their tributary. But that data doesn't always go anywhere. That knowledge doesn't go anywhere. And that's a huge issue, because how are we expected to understand the state of water in Canada if the people who know what's going on don't have that way of sharing? And so that really led us to participate in the Elevate Community-Based Water Monitoring discussion, along with Living Lakes Canada and the Gordon Foundation. And it really was about figuring out very specific recommendations for the federal government to elevate community-based monitoring. So what can be done to make sure that those community groups, those watershed groups are able to do what they do best, which is go out and monitor. So obviously I mentioned WWF Canada. We don't go out and monitor ourselves. But what we do do well is that we're a national organization. we have a very good understanding of what's going on at the national level. So we thought internally, okay, well, how can we make things better? Where can we contribute? What difference can we make as the organization that we are? And so we really see us having a role in the standardization of the collection of data. We can advocate for things like Cabin, which is amazing. The standardized way of doing things makes assessments like Watershed Reports a lot easier. We can really advocate for effective monitoring to make sure that the groups who are out there doing the work have the resources that they need to be able to do what it is that we do. And that leads into capacity building. I'll talk a bit more about it in relation to Stream, but we can support groups who maybe they have this great energy, this great willingness to do something, but maybe they just need a kick net. Maybe they just need the training, and then they'll be good to go. And that's a relatively easy solution to a problem. And again, as a national organization, we really feel like we have a role to play in the collaboration of the freshwater world at the national scale. So all of this led to WWF's participation in Stream. So here we see the different organizations in Stream and what each of us sort of do, what our responsibilities are. So I won't touch on what the University of Guelph or Environment and Climate Change Canada do, because obviously Chloe and Adam did a great job doing that just a little bit earlier. But WWF Canada's role is really to identify those regions that could really benefit from a little bit more monitoring and to work with those community groups, those watershed organizations to understand what do they need to be able to do what they do. So sometimes that's provide resources, provide training, providing equipment. Sometimes it's, okay, can we just, make sure that you can ship your samples to Guelph for analysis? What can we do to make sure that your work doesn't end up, on a spreadsheet on somebody's computer and is forgotten about? And so I wanted to end on how we work with communities. And it's a relationship that lasts far beyond just the training that we do, or we do in collaboration with Living Lakes Canada. It starts really oftentimes with outreach. We like to cast a really broad net. We want to work with as many groups as possible, and we're not particularly picky. We want to work with community-based monitoring organizations, with First Nations, with watershed groups, whoever is interested, those are our people. And so, a lot of time, it's having those calls, it's connecting over Zoom now, and to figure out, okay, what do you need? Can we work together? Do we have, something going here? And then it's figuring out, okay, now that we've decided that we want to work together, what needs to happen for that to occur? Do we need to make sure that everybody has the equipment that they need? Is it covering training fees? What needs to happen so that the monitoring can happen? And then we do the actual training. So in this case, we use the Cabin protocol, and that's where Living Lakes does their thing. I mean, it's really their area of expertise, and so Reagan is going to be talking about that. But the relationship doesn't end once the training's over. We follow up, and Stream has been going on for a couple of years now, and so we follow up with our partners for years to make sure that people still know what they're doing. Adam mentioned needing a refresher at the beginning of the season, and so that's what we're doing now, is making sure, does everybody still remember what they need to be doing? Do they need the reminder fact sheet? Do they want to hop on a call just to make sure that they know what they should be doing? Do they need to have their shipping costs covered? Maybe depending where they are, they can be collecting samples and samples and samples, but the shipping is a barrier. Is that something that we can help with? So it's a relationship that lasts so far years. And so I'll end it there. If there's any questions, happy to chat about it. And again, if you're interested in Watershed Reports, feel free to reach out. And now I'll pass it over to Reagan to bring this home.
Thank you, Catherine. Thanks so much. So I just wanted to remind everyone, if you have questions, please pop over and put them in the question and answer box. We'll get to those right after my presentation here. Yeah, and definitely check out the online tool that Catherine mentioned for the Watershed Reports. It's very handy. So thank you, Chloe, Catherine, and Adam for the introduction to STREAM and sharing your respective organizations' roles in the project. It really is a collaborative effort. So as Catherine mentioned, with the STREAM project, we are aiming to fill important data gaps for water quality across Canada. And so Living Lakes Canada's role in the STREAM project is to empower these local community-based water monitors that we've been talking so much about today. And then these community monitors can go out and they can monitor in their local watersheds. And this in turn helps fill those data gaps at a national level and across Canada. And so to do this, we recruit and train participants in a 2-day field practicum course that Adam went over. It's a modified version of the cabin protocol for those modifications he mentioned with preventing DNA contamination for the STREAM project. So my name is Reagan Mallinson, and I'm a project manager for Living Lakes Canada's environmental DNA programs, including the STREAM project. And so I've been leading cabin field training sessions since 2013 for Living Lakes Canada, and have been involved in the STREAM project since it started in 2019. And so in my presentation today, I'm just going to give an overview of what you could expect for a field practicum course in the STREAM project. And then I'll provide an overview of the participation and engagement that we've done so far in our first 2 years of the STREAM project. And then I'll close with a brief summary about the growing trends in community-based water monitoring in Canada, especially in relation to such projects as the STREAM, and how this ties into the development of the new Canada Water Agency. So Living Lakes Canada has been training cross-sector collaborators in the cabin field certification since 2013. So before the STREAM project even developed. And we were one of the first non-government organizations in Canada to go through Environment and Climate Change Canada's Train the Trainer program for the field practicum of cabin. And you can see here that a variety of backgrounds attend our certification courses, everyone from First Nation communities, non-government organizations, consulting, academia. We have professors, students, and even entire labs have attended our trainings. Industry and government as well. So as everyone has been mentioning, it's critical that the STREAM trainees learn how to collect the data in a standardized way. And this is why the cabin protocol is so great. And so everyone across Canada is using the protocol, collecting data in the same way, using the same equipment, and has the same knowledge of why that needs to be done that way. And so this allows the network to share data on the open access cabin database that Adam talked about and who Emily will talk about on our next webinar series. And so then users of the cabin database understand that what this data means and also that they can trust it. So as mentioned many times, cabin is a method of biomonitoring, which uses the bugs on the bottom of the stream to assess water health. So participants can then begin to answer their own watershed health questions, such as, how successful was my streamside restoration? Or is there fish habitat, or is there suitable fish habitat in this river? Or what is the general health of my river? So in order to understand the habitat the benthics are living in, we collect a handful of metadata. So this is really why we need that 2 days for the cabin field practicum. And this includes water chemistry, channel measurements, reach characteristics such as surrounding vegetation, and substrate data. And participants learn how to collect the benthics via a kicknet method that you can see on the screen here. And it looks pretty nifty when they're in the water. It looks like a bit of a river dance. So it's pretty fun doing that with everyone in the rivers. And so this metadata really helps us interpret the results to relatively assess how healthy the streams are by comparing what kind of benthic biodiversity we expect to find and what we actually are collecting. So once we've collected these bug samples in the river, we then pack them up carefully, gloves on, to minimize contamination, as you can see. We pack them up carefully and ship them to the University of Guelph for processing via the DNA metabarcoding that Chloe talked about today. And so many of our participants, along with the DNA samples that are collected as first stream, are collecting the morphological samples. And this really benefits them because then they're able to use the existing cabin analysis and reporting tools. And Adam went through all that. So here is a brief overview of what participants can expect from a 2-day field practicum as part of the STREAM project. And so before the course even starts, as Catherine mentioned, like we have had plenty of planning meetings. We've done desktop exercises to identify potential training and monitoring sites. And what we're looking for in these training and monitoring sites is, first of all, like how big is the stream? We really want to make sure it's wadeable. So, you know, participants don't go floating down the stream. Another is the ease of access of the site. So can we drive to it? Can we get down to it? Like, want to make sure like the stream side isn't thick with blackberry bushes, for instance. And then we also want to make sure that there is a suitable amount of shoreline available. Our groups usually hover around the 10 before COVID, 10 people per training. So we need enough room for people to move comfortably around, and there's quite a bit of equipment that go along with this protocol, so room for that as well. And so the Living Lakes Canada team and WWF, when they join us, arrives usually 1 to 2 days early before the official start of the field practicum. And this really helps us build relationships with the local communities. We go out to the sites to make sure that everything looks okay. You know, there hasn't been a major rainstorm, sites are accessible, they're appropriate, etc. We can do a lot of this via desktop exercise, but that double check is always really important. And so then the official 2-day field practicum kicks off by a roundtable of introductions, again, just like allowing everyone to get to know each other, past experience with monitoring, if you've ever used cabin before. Then we introduce the STREAM project, how it's similar and related but different from the traditional cabin field certification. And then we launch into health and safety, which in the past has just been safe wading protocols, but now includes quite extensive COVID protocols, as you can imagine. So for the STREAM project, our field practicum, we use a modified version of the cabin protocol. And this really just includes like the pre-monitoring decontamination of all the equipment. And why we want to do this is because we want to make sure that the DNA that we're sending to Guelph is from the site that we're at, and it's not being carried over from any of the other sites or storage lockers or back of the car where the equipment could have been before. So everyone participates in the decontamination of their waders and supports with the equipment decontamination, and we follow the standard operating procedures that ECCC created. So it's really just an easy modification to the cabin protocol. And so day one includes a demonstration. Once we actually launch into the field certification, day one includes a demonstration in the morning of all the equipment and how it's used and what it's used for and why. And then we let each participant practice with it. There's quite a few steps, so repetition is definitely key. So the whole next morning, we allow participants to apply the skills that they've learned the previous day on a new habitat. And this really builds problem-solving skills. They might have questions in a new area that they didn't have before. So that new environment is key to build that confidence. The afternoon of day two is the certification. So essentially, it's just like another round of practice, except for the trainers aren't supporting or giving any advice, and we just provide feedback to all the students at the end. So what's been great as part of the STREAM project, what we've been able to do a bit differently is we've been able to stay a couple days after that official 2-day field certification course and go out to the local groups monitoring sites instead of just being at the training sites. And then this really allows groups to practice again the skills they've learned. We really let them take the lead, so it builds confidence and it builds that memory, so that, you know, when they haven't gone out for a year over the winter, that when they come back to it, they've done it so many times that it's that the knowledge is just there. But of course, everyone gets a little rusty over the winter, so we will be providing refresher courses for those that are interested, as well as please check out the operating procedures that ECCC has created. So now that you have like an overview of what we're doing out in the field, let's check out where we've been so far. So as Catherine mentioned, we prioritize watersheds where we lead trainings in. We wish we could be everywhere at once, but obviously that's not possible. So we focus first on the benthic deficient areas. And once we've identified those, we also focus on the capacity and needs of the local organizations we partner with. So other groups that are on the ground that need and want training. And then we also focus on match funding to support the development and sustainability of local biomonitoring programs. So do groups have their own funding that they can really make a long-lasting program? So in 2019, we were able to lead 2 field certifications in the Columbia Basin, in Nelson and Invermere, in BC, the Skeena Basin in Smithers, and this is in partnership with a few nations from the Environmental Stewardship Initiative. And then also in the Peace Athabasca Basin in BC, just north of Fort St. John, with the Blueberry River First Nation, and in Alberta, in Canmore, Alberta, with a variety of groups there. So in 2020, which was year 2 of the STREAM project, we were faced with many challenges like everyone else due to COVID. But Living Lakes so we had originally planned to be in Ottawa, Thunder Bay, Kenora, all in Ontario, and Whitehorse in the Yukon. But of course, and unfortunately, due to travel restrictions and community closures, these were all postponed. So as travel restrictions began to lift late last summer, we were able to adapt the modified cabin protocol to include appropriate social distancing, personal protective wear. So on top of all the gloves and equipment that we're wearing for decontamination, we also had masks, sanitization stations available, and made those smaller groups for participants. And so instead, we were able to hold a course in the Old Man Watershed in Coleman, Alberta, and this was in partnership with the Old Man Watershed Council and a number of WPAC representatives that attended there. And so we were also able to carry out our originally planned BC-based courses, including in Nelson, BC again, this time in partnership with the Friends of Kootenay Lake Stewardship Society. And then also in the Nechako watershed with the Binche-Witten First Nation. And so 2020 really was able to show the resiliency of community-based water monitors and what they can achieve. Government and industry all had to shut down their long-term water monitoring programs because of the pandemic, but the community monitors were still able to get out and collect that data. Sorry. So yeah, where will we be next? So we have, we're going to prioritize the courses that we weren't able to do last year. So the 3 in Ottawa and the 1 in the Yukon, and we'll see if those are able to go through, and this all depends on COVID restrictions, obviously. We also have 1 planned for Rocky Mountain House, the Peace Athabasca, again with the Blueberry River First Nation. They need more capacity building. So that really just shows like what Catherine was mentioning about the relationship building. We're not parachuting in, we're coming back to support those organizations that need it. We also have a local BC-based course in partnership with the Secwepemcúa Restoration and Stewardship Society. So at this point right now, we have quite a few courses on our roster, but we are starting a wait list in case there are areas that we can't go because of travel restrictions. So yeah, that wait list has been started. So far through STREAM, Living Lakes Canada and our partners have trained over 100 community-based monitors in 10 communities across Canada. And so the community-based water monitoring, or CBWM, movement that is growing through projects like STREAM is one that Living Lakes Canada has been working to mainstream since our organization was founded. And in a national survey coordinated in 2017 with Living Lakes Canada and SFU, the Simon Fraser University, and the University of Acadia, to see what groups were doing across the country, we found that there had been an exponential growth of community-based water monitoring over 10 years. So based on that information, we joined forces with WWF Canada and the Gordon Foundation to convene the National Roundtable discussion around community-based monitoring. And this was attended by 50 Indigenous and non-Indigenous community-based water monitoring leaders in Canada. And it was convened 1 year before the federal government mandated the creation of the Canada Water Agency. So the roundtable has made 60 recommendations that were distilled down to 4 key priority actions. And so the Roundtable Coalition has submitted these 4 priority actions as part of the public consultation process to inform the newly developing Canada Water Agency. So we hope that the federal government will take these actionable steps to show leadership and support in advancing community-based water monitoring of freshwater ecosystems in Canada. These are unprecedented times, and the biodiversity and climate crisis will require shifting paradigms with all hands on deck for collaborative problem solving needed. So thank you everyone so much for your time and attention. We really appreciate it. If you have any more questions, please feel free to reach out to me. My contact is on the slide there. So I'll stop screen sharing and then we can launch into the question and answer session. And sorry that we've left it a little short, but maybe we can go a little over the hour. So it looks like it's going off. So everyone's up here. So I'll just start from the beginning. Again, just want to remind everyone that there have been some links posted in the chat section, so definitely check those out. So Carly Simpson says, is the metabarcoding using water samples or individuals or both? Yeah, this is a question we get a lot. So I know Chloe, you covered it in your presentation, but maybe do you wanna tackle that question again?
Yeah, sure. So for stream, we use the bulk tissue samples. So these happen to also contain the actual individual bugs themselves. We use through kick netting, and we take that sort of bulk sediment sample and we essentially put this in a blender and take subsamples and extract DNA directly from the benthos. So we do not do water sampling for stream. That is one way to target eDNA, but actually in our lab we did some paired testing with benthics and with water to look specifically at invertebrates, and we found that the best sort of representation of biodiversity is using the benthos. So yes, we use benthic sampling, not water sampling for stream.
Awesome, thank you, Chloe. So Anwin says, how does WWF determine what is data deficient and what isn't?
Yeah, that's a good question. So really what we look at is there enough data in recent years? We don't want to be looking at data from 30 years ago and assuming that if it was good 30 years ago, it's good now. So we want recent data, and we want a good geographic diversity of data within the subwatershed itself. So these are national assessments, the Watershed Reports. So they look at a very large scale. And so the areas that we cover are quite big. But yeah, so we want to make sure that we've got temporal representation and geographic representation as well.
I think Reagan has temporarily frozen. So yeah, I can go ahead and continue with questions, I think, until Reagan manages to get back on. So Carol Doyle asks, have any studies been done on Charman Creek in Gibsons? And if so, could I see the results of the study? If not, can I be trained to have one done? So personally for me, I've not seen any samples come in from this area. I don't know if Adam or Catherine, if you're aware of any samples off the top of your head. Obviously not, we don't know the entire database on the top of our head, but we can always follow up.
Yeah, I know we did do, this was a few years ago now, just took a couple samples in Gibsons or around there. I don't remember if it was that creek or not. Oh, Regan's back. But that was, before stream, that was just to sort of test out a methodology. So there's no report for that. And so no reports. And if you are interested in getting training, please reach out to Reagan, and then we'll figure out what to do from there.
Sorry, everyone, I don't know what happened there. Thanks for stepping in, Catherine. So sorry, I was in the middle of Aaron Beer. So is it able to do abundance? Is the technology effective in detecting species at low population abundance if there is plentiful DNA from high abundant species present? So Chloe or Adam, who wants to take this?
Sounds
Yeah,
like
so
a question.
yeah, it's a really good question. So that's why it's so beneficial if groups are already collecting morphological samples to continue to do this, because at the moment with DNA metabarcoding, it is not possible to get a reliable estimate of abundance. What we can do, however, with metabarcoding is we can look at proportions of reads. So essentially you have so many sequences produced, and you have so many replicates of those sequences, which we class as reads. So we can look at read abundance, which in some cases has been seen as a proxy for abundance. However, we don't often recommend this because it's not true abundance. But the technologies are developing, and potentially one day DNA will be able to give a more reliable estimate of abundance. But that's why at the moment, if your already existing sample project is looking at morphology, that's why we recommend to combine the two and add stream on as an additional method. But no, not quite yet at the moment, but we have ways of representing the data to give an estimate of read abundance.
Mm-hmm. And there's just a question later on, I'll just answer right now because it's kind of related, but this question is, does metadNA barcoding also produce abundance data? And if it does not, has Stream developed a new analysis method in place of traditional indices? So yes, we are in development of new indices right now, which are only use presence and absence data in terms of the taxonomy. And yeah, we'll have more information on that as they become available.
Great, thank you both. So maybe another one for Chloe. Can the eDNA be used to identify fish species in a watershed? Example, Atlantic salmon. If so, can it be used to identify the particular population?
Yeah, another great question. So yes, of course, this method can be applied to basically any taxa that lives in freshwater systems, and obviously terrestrial, but we focus on freshwater. The Stream project itself does not have the capacity to test for fish as well. But if this is something that a group has funding for and would like to do some analysis on fish, then that is definitely something we can set up with the lab, because the lab I work in also sets up contracts with community groups to analyze samples. So yes, most definitely we can do both. We focus on metabarcoding, but it can detect species across the system, and we can also run some analyses looking at a little bit of genetic diversity too. So we run some haplotype analysis. So yes, you can do both species detection and you can look at whole populations.
Awesome, thanks, Chloe. And then from Anwin, what are the additions to the Stream protocol which aren't in the regular protocol?
I can take this one. So I think you mentioned a lot of it actually in your presentation, but some of the main differences are actually decontaminating your own equipment and your bodies before collecting the sample. As you can imagine, going from stream to stream, you're probably carrying debris on your boots and on your waders and your kick net. So we want to make sure that that's all really well decontaminated with a mild bleach solution. And then also afterwards, preserving your sample. It is just recommended to use ethanol, like alcohol, a very high percentage of alcohol, in order to preserve your samples for Guelph. But more recently, they just had a project where you can use a specific kind of antifreeze if you do have issues obtaining ethanol where you're located. So if you're interested in that, Chloe can provide some more information on it.
That's fantastic. Thanks, Adam. So Alan James says, our groups has around 20 years of data using the Stream Keeper's Handbook, Module 4, Stream Invertebrate Survey. Is there a way to convert this data to cabin? Adam, do you want to take this one too?
I am actually not familiar with the Stream Keeper's Handbook, Module 4.
Me neither.
Potentially, yeah, I can't really answer that question without some background on what the handbook is actually.
Okay, we will answer that question in our follow-up email. So are there any ways that this data is used to protect streams from, where did that just go? Protect streams from pollution or anthropomorphic degradation? What are some examples of these results being used for stream remediation? So I can take this one because many of our different community-based monitoring groups have different research questions that they're asking. So the traditional cabin analysis tools give you a grade of how different your river is from what's expected. So with the DNA, the analysis tools aren't developed yet. So right now we're just getting a biodiversity of the benthics present. So there has been, and please tune into our other webinars because there has been examples of our partners performing streamside restoration to prevent off-road vehicles entering the stream. And they are monitoring how the river is, like how healthy the river is compared to before. So yeah, please tune into our other webinars for more information about how the project is being applied. Does anyone else want to comment on anything there?
Yeah, I mean, we can just say that one of the really cool things I think about Stream is that you know, it's a tool, right? The groups and the nations and, you know, the watershed groups that we work with, they're the ones who come up with the research questions. So really the sky's the limit in terms of what this can be used for. And so it's really up to the groups if there's a particular concern. So you mentioned the off-road vehicles, if there's a concern with salt contamination from the winter, if there's, you know, it could be, there's so many things that this could be used for. So it really depends on the research questions that the individual groups have.
Awesome. Thanks, Catherine. So I see that Kalish Fraser asked for the SOP, so we can send that out in the resource package as well. So then Dennis says, I'm planning a canoe trip on the Peace River this summer through BC and Alberta, and it just occurred to me that it may be a good chance to do some sampling in isolated areas. If you're interested, perhaps we can get something up. So yeah, please reach out to us, Dennis. Be great to follow up and hear more information and see if that could work. Date for BC course. So this is a closed course with the Secwepemcpool Restoration Society because they have quite a few people that need training. It is scheduled for the first week of May. We'll see if that's realistic. But yeah, please keep posted to our website. We do have to keep our courses smaller right now because of COVID. So there is a wait list. If you're interested, please in being added to the wait list, please send us an email. Okay, so Brendan says, this might be a bit technical for this webinar, but traditional morphological based biomonitoring relies on abundance as well as presence absence. Does metabarcoding also produce abundance data? Has Stream developed new analysis methods in place of traditional indices? So, oh, sorry, Adam, you answered that one. Yeah, whoops. And then Shelby says, are other bioindicator groups included within the Stream project? So, algae and microbial, if not, is branching out to other communities something that you're looking into? Chloe?
So yeah, at the moment, the Stream project, because we're obviously limited for funding, we only focus on benthic macroinvertebrates. However, as part of my postdoc at the University of Guelph, we are currently doing a study looking at combined diatoms and benthic macroinvertebrates and their ability to perform as bioindicators together in DNA samples. This is something that won't be included in the current Stream, but any sort of extension of the project or future work which is still in the works, we don't have a clear idea of where the project is going beyond its current timeline, that is definitely something that we'd be interested in. And the great thing is you don't have to do anything different, because we get that diatom information from that same KitNext sample. So it isn't something so much that community groups have to do in terms of more than just their sample. It's more of an analytical process.
Great. I think that also addressed the next question from Sophie about if there's going to be support from shipping and DNA analysis into the future years. We just aren't at a point to really know right now. So stay tuned. From Phil, have you had much success in finding invasive species or other rare endangered species using DNA, using metabarcoding? Chloe, I'm wondering if you can talk to the whirling disease example.
Yes. So in terms of, there have been, again, as what Catherine said, a lot of the time when I do extra scans of the data, it's mostly based on the community's questions. So I've had a couple of community groups ask me to look for a particular invasive species in their data set, and I haven't found, on both occasions, I didn't find the invasive species. So this is very much on a community-driven question. So the taxa reports generated, the bioindicator groups are highlighted, and the reports are sent back unless any additional analysis is requested. However, for example, the Ghost, correct me if I'm wrong, Reagan, but the Ghost Watershed Alliance Society requested a little bit of interpretation for the sludge worms, which are hosts for whirling disease. And so in their report, I made sure to take note of exactly where that species was across the sample sites, and added an extra paragraph of interpretation of what this could mean for either potential spread or containment of whirling disease within the catchment. So again, these are very much community-driven questions. There is a sort of standard reporting process that comes with the stream reports, unless any additional information is requested when samples are submitted to us.
Fabulous. Thank you so much, Chloe. So we'll just do a couple more, and then we can tackle the rest of the ones offline in the follow-up package. So Cody Johnson says, does the testing indicate the presence? I guess you just answered that. So I'll go down to Rob Knight's question. Can you use a previous cabin samples for DNA analysis? Contamination would be an issue, I guess. Good question. We get this question a lot. Yeah. No, you can't. Yeah. Or Adam, do you want to take it?
No, yeah. DNA contamination is obviously the primary concern. Like, you would never know whether or not there's been previous DNA in that sample, and also preservative is also an issue as well. If there's been any contact with formalin, I know that's not ideal. So yeah, unfortunately, we can't use previous cabin samples.
Okay, great. And then, yes, Diane, please email me if you want to be added to the wait list. Thank you for the link, Jerry. And then the last question that we'll answer today, the last question is, has the stream approach been applied to lake invertebrate samples? So Chloe.
Also a really good question. I'm liking these questions. So yeah, we've actually just completed a pilot study in conjunction with Environment Canada on collecting benthic samples from Lake Ontario. So this is something that we're looking to push into. This again won't be part of stream, because stream is mostly focused on rivers and wetlands. But this is something that we're looking to branch into, and it is not widely being applied at the moment. There was a study in 2018 comparing morphology with DNA metabarcoding samples, and we're building on this study. We're hoping to get a publication out soon, so we can keep you in the loop with that. But this is definitely a direction that we're looking at heading towards. Great.
Thank you so much. So that wraps up our question and answer session and our first webinar of the stream series. So I just want to give a huge thank you to all our speakers, Chloe, Adam, Martin, thank you, and Catherine. Big thank you to you all, and thank you to all our participants who attended. We really appreciate your interest and your time and attention in the stream project. So thank you so much. Everyone have a great day, and see you again soon.